Hi everyone. I want to talk a little bit about the common kinds of media that we use in the microbiology lab. Media is just the food that the bacteria eats, and there's hundreds of different kinds of media depending on what kind of bacteria you want to grow or what kind of fungi you want to grow and the specialized conditions. A general purpose medium that we use frequently in the lab is called nutrient agar or nutrient broth. Now, we can have this medium in a liquid form, and here we have a nutrient broth tube, and there's just a little bit of liquid in here. Or we could have it in a solid form, and here's a nutrient agar plate. So it turns out these are both have the same ingredients, except the media in this container has something in it called agar. Agar is a solidifying agent. If I introduce bacteria into my nutrient broth, the bacteria will grow, overnight or within a couple days, and they'll all be mixed up in there. And typically we would use that culture relatively quickly, within a day or so, because it could all dry up. Okay, so nutrient broth, I want to grow a little bit of bacteria. I don't care if they're mixed up, and we would use it within a couple of days. Nutrient agar can be used in a little bit different way. People wanted to be able to take different mixtures of bacteria and spread them out so the bacteria are far enough from each other that each one will grow up separately. And when we grow bacteria from a single cell, we call it a colony. And so you can end up getting colonies of bacteria on here, and you can separate out different types of bacteria. So nutrient agar plates can be used to grow bacteria in colonies. Also, they tend to last a little bit longer here on the agar plate. So if I want to keep some bacteria for about a week or so, I might grow it on a nutrient agar plate. Fortunately, bacteria don't degrade agar very well. They don't use agar as a food. So even when the bacteria start growing on here, this medium will remain solid. Sometimes we can take that nutrient agar and put it in a screw cap test tube. And you can see here that this tube has some agar in it, but it's kind of at an angle. When I make the agar, I have to heat it up until it's boiling, so it's in liquid form. And then I can pour it in here and cool it at an angle, and then it will solidify as it cools. And I have what I call an agar slant. Now, because I have a screw cap tube on here, screw cap lid, if I grow bacteria in my screw cap tube on my agar slant, the bacteria tend to last longer. So I'll grow it up overnight, and then I'll seal the lid and put it in the refrigerator. And most bacteria will last for a couple months on these agar slants because they won't dry out. The last term I want to introduce to you is, here we said this was a nutrient agar plate, but a lot of people get the term Petri plate and an agar plate mixed up. The Petri plate, invented by a guy named Petri in the golden age of microbiology, is actually the dish, the empty dish that's used to create agar plates. So again, I would make this agar media in the laboratory. I would sterilize it, pour it in here while it's still in liquid form, let it solidify. So I have the Petri plate, or the Petri dish is the empty plate, and now I have a nutrient agar plate. I have nutrient agar inside of a Petri plate. Got it? Hope you understand a little bit more about media.